Okay, let's get started. Welcome to the webinar Overcoming Barriers in Drug Metabolism, the HEPATOPAC Reaction Phenotyping Models for Low Turnover Compounds. This is the last in a series of 5 webinars in which we are talking about novel applications for HEPATOPAC. I am Seema Ranjan and I'll be facilitating the webinar today. I'm the Director of Facilities and Instrumentation at BioIVT's lab in Kansas City, the legacy Xenotech site where we offer a comprehensive portfolio of in vitro ADME research services and products. Before I introduce my colleague Jeannemarie Gaffney, our speaker on today's webinar, I would like to take a few minutes to tell you about BioIVT. Founded in 1981, BioIVT has grown to become the largest provider of biospecimens and research services to the pharmaceutical and diagnostic industry. We are dedicated to providing the services and biospecimens that our customers need to help them advance their discovery, preclinical, and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and become the preferred provider for our global biopharmaceutical clients. As a legacy Xenotech scientist, I want to highlight that Xenotech joined the BioIVT family in 2022, bringing its storied reputation for ADME research services into BioIVT. BioIVT has continued to invest in and expand our service capabilities. The acquisition enabled us in the ADME services group to access BioIVT's extensive portfolio of biospecimens. BioIVT and Xenotech share a similar culture of scientific excellence and passion for helping our clients achieve their research goals, which makes bringing our companies together a great fit. I lead the analytical services at our Kansas City facility and have over 15 years experience working with my colleagues to design and implement ADME programs that comply with the most up-to-date regulatory guidance and help them achieve their research objectives. One of our unique strengths is that we partner with our clients to design the studies they need. When conventional methodologies might be difficult, might not be sufficient to answer our clients' questions, we can employ custom techniques to answer their specific research questions. For example, we can utilize a long-term HEPATOPAC model in our studies to evaluate low turnover and slowly transported compounds. At the foundation of everything we do is a steadfast commitment to ethical sourcing, uncompromising quality, and a genuine dedication to partnering with the research community. We take pride in helping our clients find solutions for their difficult questions. Here is a snapshot of the area of BioIVT's portfolio, including products used in cell and gene therapy programs, normal and disease state biospecimens, and products and services to evaluate the ADME properties of drugs and prepare for IND submissions. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts works routinely with clients to help them identify hepatocyte loss, having the characteristics needed to meet their specific requirements. On the ADME services side, we work with the client to design and conduct IND-enabling packages, including enzyme induction, inhibition, metabolism, drug transporter, as well as other studies to predict the pharmacokinetics of drug candidates. Our analytical team is experienced in small and mid molecules and develops custom methods to support these studies, as well as offers in vitro metabolism services. Our ADME services group provides individual studies as well as integrated programs based on our clients' needs and challenges. Many of our clients start with a consulting engagement in which we provide expert advice to design ADME programs, assist with gap analysis, analyze existing data packages, or help prepare for regulatory meetings. We offer full in vitro ADME programs at our labs in Kansas City, Santa Clara, and Durham, North Carolina. When clients choose to work with us, they get the benefit of a team of experts who collectively have worked on thousands of programs and bring that experience to each new project. If you're interested in or have questions about analytical or metabolite ID services, I'd be happy to talk to you, but that will have to wait until another time. Additionally, we have partnerships that enable us to integrate in vivo studies into preclinical programs, and we have extensive capabilities for bioanalysis to support these programs or as standalone cells. You can also visit BioIVT.com to learn more about a portfolio of products and services. Now it is my pleasure to introduce Jeannemarie Gaffney. Jeannemarie's research efforts have focused on the development of tissue-engineered products and cell-based in vitro models with a focus on primary mammalian cell culture. In 2009, she joined Dr. Salmon Khetani's team at Ascendance BioTechnology when they were in the early stages of developing micropatterned co-cultures, including human, rat, mouse, monkey, and dog hepatocyte models, a system later known as HEPATOPAC. As the product was commercialized, she supported all aspects of producing HEPATOPAC kits to meet growing customer demand, which continued after BioIVT acquired the technology in 2018. At BioIVT, she also provides technical assistance to our clients who are using HEPATOPAC cultures in their drug discovery and development programs. Jeannemarie, I'm looking forward to your presentation.
Seema, are you able to see the presentation?
Yes, I am.
Great, thank you for confirming. Thank you again, Seema, for the introduction today. Today, we're going to discuss reaction phenotyping methods for low turnover compounds, specifically in our HEPATOPAC model. What is HEPATOPAC? HEPATOPAC is a long-term, stable, reproducible, and more predictive platform. This platform was developed at MIT in the lab of Sangeeta Bhatia and Salmon Khetani. This is a mature and well-established model. Their initial work utilized microfabrication tools adapted from the semiconductor industry. This has allowed us to create domains of hepatocytes surrounded by stromal cells. In the bottom image here on the left are examples of those domains of hepatocytes which have been stained in the wells. The image here on the right provides a representation of the islands of hepatocytes surrounded by the stromal cells. This micropatterned architecture allows for homotypic interactions between the hepatocytes on these islands and heterotypic interactions between the hepatocytes and the stromal cells. This allows for optimized cell-cell, cell matrix, and soluble factor interactions. In addition to using this micropattern technology, we are able to further optimize the architecture. We're able to control the cell-cell interactions using this microfabrication technology. This is an example here of different size islands that could be created. The one here on the far right is closest to our optimal architecture that we do use of 500 microns in most of our HEPATOPAC models. In addition, we're able to control the layout of these islands. They're specifically earmarked so that they are 1200 microns center to center of these islands, and this is consistent throughout the wells. Looking a little bit at functionality and longevity, here are examples of albumin secretion and urea synthesis. Hepatocytes remain healthy and functional for several weeks in HEPATOPAC. These data here are specifically for 28 days of culture, and as you can see, the albumin maintains a stable, steady secretion level over the course of this study. Here for the urea, once again, we see steady urea synthesis in the HEPATOPAC model. The line here represents 2D culture, which you would start to see at the end of the first week, a reduction in the function of those cultures. And we do see high levels of function in human and also preclinical species. Looking at one of the functional markers that would be important for our discussion today for reaction phenotyping would be the P450 enzyme activity, phase 1 and phase 2. These are stable across several weeks. These cultures here were monitored out once again for a 28-day period. The cultures are seeded on day 0, and you can see in the first week of culture, the cells are recovering in our co-culture system, and then they reach a steady state and maintain that function over a multi-week period. Over here, you can see, as I mentioned, it does take about a week for the cultures to mature. So here's an example of HEPATOPAC at day 8 compared to suspension. Freshly isolated hepatocytes in suspension would be considered a gold standard for hepatocyte function. And the HEPATOPAC model at its mature state does perform comparable to hepatocytes in suspension. Why is long-term HEPATOPAC function important? It allows time for establishment of disease models in vitro. It enables us to do repeat dosing experimental timelines. And specifically for today's conversation, it does support pharmacokinetic assessment of low turnover compounds. I did just want to point out two different assays for standard clearance generation. On the left would be a standard clearance time points, and you can see on the x-axis, these are in minutes. So this was a 60-minute assay, and you can see after 30 minutes, that parent compound has been completely depleted. On the right-hand side is a HEPATOPAC clearance study. And you can note here on the x-axis, this study was run in an hour's time frame. And specifically, this study ran out for 168 hours or 7 days. And you can see we were able to see depletion of this compound. If this is a low turnover compound, if the same low turnover compound was done in only a 60-minute assay, we would not have been able to measure any depletion. So depending on your compounds, there can be different assays that may be best suiting for your needs. In addition to maintaining metabolic competency and differentiated function, the hepatocytes exhibit quality morphology in the HEPATOPAC model. There's characteristic polygonal morphology, distinct nuclei, biocanaliculi, and visible cell-cell junctions. And we do see this in human and preclinical species. Here are some examples of biocanaliculi staining with the CDC FDA stain, and also MRP2 protein staining. Touching on some of the applications for HEPATOPAC, initially HEPATOPAC was used for a lot of work with small molecules, and the work focused in the metabolism area, such as MET-ID and MET stability, and also included work for toxicity. The long-term culture period allowed for a repeat dosing regimen. Additionally, work has been done in the liver disease space as well, with HBV work and malaria work. The long-term culture multi-week period did allow for these types of disease models to be tested. More recently, we have done work with steatosis modeling, and my colleague Carissa recently presented in a previous webinar on our steatosis modeling. The HEPATOPAC model is able to load lipids. And my colleague Scott recently presented a webinar on HEPATOPAC specifically for siRNA transfection, GalNAc conjugated siRNAs and oligos via free uptake. And we have now been pairing our siRNA work and our GalNAc work with some of these different applications, as you saw recently in Carissa's presentation for steatosis modeling. Today, we're going to be discussing some of our siRNA work coupled with MET stability for reaction phenotyping. And just quickly, as a refresher, HEPATOPAC is available in human and preclinical species. For human, we offer single donor and liver pool options. For rat, we offer the Sprague-Dawley and Wistar-Hahn strains. For monkey, we offer Cyno and Rhesus. For dog, we offer Beagle Dog, and we do use freshly isolated hepatocytes. And for mouse, we offer both CD1 and C57 strains. And this platform is available in a 96-well, 24-well, and a 96-well high-content imaging plate. And any work with HEPATOPAC can certainly be outsourced to BioIVT as part of our contract research services offerings, or the plates may be purchased for use in your labs. And before we move on to the reaction phenotyping, I did want to point out that our website does have resources for other applications which you may be interested in as well. And specifically, there is a publication compendium which lists a number of publications and also indicates what type of application it might be. Toxicity, metabolism, steatosis, so it may help you find the information that you're looking for. So I first just wanted to touch on clearance in the HEPATOPAC model, which will be the basis for allowing us to run reaction phenotyping studies. And what is metabolic stability or clearance? The goal of this type of assay allows us to measure how efficiently the liver extracts a drug from plasma and eliminates it from the body. Why are clearance studies important? It helps determine a dosing regimen for the drug and to make sure the plasma concentration of the drug is within the therapeutic or efficacious range. how are clearance studies conducted currently? Suspension cultures, recombinant enzymes, plated monolayer of hepatocytes, or next-gen models such as HEPATOPAC. Today, we are going to discuss HEPATOPAC, but if you have needs with suspension, recombinant enzyme assays for clearance, our group here in our contract services would be happy to speak further with you. And so why would you use HEPATOPAC? Specifically, since it does have maintains metabolism over a length of time, we are able to determine clearance specifically for low turnover compounds. And typically, a clearance study in HEPATOPAC lasts approximately 7 days. And to provide you a little bit of background on predicting clearance in HEPATOPAC, this is a publication I'd like to refer you to from a group at Sanofi, where they looked at 15 different low turnover compounds. Specifically, they looked at 3 different models: suspension, monolayer, and HEPATOPAC. And they used the same donor for all 3 models. And they were looking at the predicted clearance in vitro versus the observed in vivo clearance. And 46% and 38% of the compounds, respectively, in suspension and monolayer predicted accurately within 3-fold. And the HEPATOPAC model predicted accurately within 93% of the compounds within 3-fold. Similarly, this would be a reference from a team at Roche, where they looked at 36 marketed compounds. And once again, they were predicting hepatic clearance, and it was accurately predicted for 34 of the 36 compounds within 2- to 3-fold error. Now that we've established that you can run low turnover clearance studies in HEPATOPAC, we're able to move on to reaction phenotyping of low turnover compounds. Compounds with a single route of elimination have a high object drug potential. Therefore, studies to identify enzymes involved in the metabolism of a drug's clearance are essential to understanding its DDI potential. Traditional methods for reaction phenotyping don't work well for low turnover compounds. Those typical methods, as I mentioned before, might be microsomes with specific enzyme inhibitors or recombinant enzymes. Repeat dosing with specific chemical inhibitors can be done in HEPATOPAC. And there are some great publications which are available demonstrating this, specifically from some teams at Roche and at Merck. However, this is a labor-intensive activity, and it does not allow for assessment of enzymes which lack a specific chemical inhibitor. Therefore, we've been looking to combine the high-quality clearance prediction of HEPATOPAC with the ease of use and customization potential of using siRNA to run low turnover reaction phenotyping studies in HEPATOPAC and to make this easier. Now we'll move on to touching on the gene knockdown in HEPATOPAC specifically for reaction phenotyping. In some of our previous webinars, my colleagues did touch on how the gene knockdown has been used in disease modeling and therapeutic approaches as well. But today we'll specifically talk about ADME studies. And our objective in developing an effective siRNA transfection method in HEPATOPAC involved a hepatocyte-specific transfection, the use of commercially available transfection reagents, and maximizing the durability and reducing toxicity. And we did know, obviously, that there's potential applications for using siRNA in HEPATOPAC. We already had work with clients doing research in the hepatocellular mechanisms of disease and RNA-based therapeutics. So our focus then turned to trying to apply this same model and to compare it with chemical inhibitors for reaction phenotyping. In one of the previous webinars, my colleague Scott did provide a lot of background on siRNA knockdown in the HEPATOPAC model using transfection, GalNAc-free uptake, and I just wanted to touch again on how we are doing this specifically for reaction phenotyping. Hepatocytes in the HEPATOPAC model are seeded on day 0 of culture. On day 1 of culture, the stromal cells are added to initiate the co-culture system. It then takes about a week for the cultures to mature, and then they could be used for a study, either here at BioIVT in our services program or shipped to the lab of a customer for use on their end. When we were doing transfection for the reaction phenotyping, we want to be using targeting just the hepatocytes. So that siRNA knockdown occurs on day 0 of culture when just the hepatocytes are present. This is an example of some of our early work targeting 3A4. And we are using Dharmakon smart pools typically for our siRNA knockdowns for reaction phenotyping, and we're using commercially available RNA max as our transfection reagent. Our initial studies were looking at CYP activity via mass spec, and then also looking at mRNA levels. And you can see with both of these methods, we were showing an 80% knockdown in our early studies for 3A4. So once we had our initial work completed, we then moved forward with some larger scale and repeat studies. So we were looking at the reaction phenotyping for the FDA recommended CYP450 enzymes. And we looked at 1A2, 2B6, 3A4, 2C19, 2C8, 2C9, and 2D6. We used siRNA to knock down each individual gene. And as I just mentioned, this transfection was done on day 0 of culture with just the hepatocytes present. We then collected cell lysates on day 9 and day 15 to assess gene knockdown by PCR. We chose these time points because they would be considered the beginning and the end of a clearance study. And when we look at this in summary for the genes, at day 9, we see 65 to 95% knockdown of the genes. At day 16, we're seeing 55 to 85% knockdown of these genes. In addition to looking at mRNA, we were also going to assess whether the knockdown was selective for the single enzyme isoform that we are selecting, and to look not just at on-target knockdown, but also off-target knockdown. We used typical probe substrates to assess metabolite formation by LC-MS. We did matrix all siRNAs with all substrates. And as I mentioned, these were 3 separate studies in order to look at some repeat data and reproducible data. And here would be a slide with the data from that specific metabolic activity assessment of on and off-target knockdown. In blue, I will highlight for you would be the targeted knockdown. So this here would show the knockdown of 2C9. Knockdown by activity ranged from 63 to 80% on day 9 of culture and 52 to 82% by day 15 of culture. We did set a threshold for off-target activity at a fold change greater than 1.5 or less than or equal to 0.5 compared to the scramble control. And these are highlighted in red. So you can see there is some off-target effect, but it is minimal. There is a lot of data provided on this slide, and as Seema mentioned, these slides will be shared. So you will be able to review this data in more detail after the webinar. So obviously, one of our key questions was to see how siRNA inhibition compares to chemical inhibition. So we specifically looked at 2 different targets, CYP3A and CYP2D6, and the selective inhibitors that we used were azamulin and paroxetine. When we're doing chemical inhibition, we actually seed the cultures just like we would for gene knockdown, but once the cultures are mature is when we start to add those chemical inhibitors. So that would be right before we're going to start doing our clearance study. When we're doing chemical inhibition, such as azamulin, we need to dose every day with that inhibitor in order to maintain suppression of that CYP. For paroxetine, we need to redose that inhibitor every other day in order to maintain that suppressed CYP activity. And similar to our gene knockdown method, we once again assessed on and off target fold change using metabolic activity. And you can see here in blue, once again, shows the activity suppression, which worked very well for the chemical inhibition. In red, we do have highlighted some fold change, which we did see for some of the off-target effects using this for the chemical inhibition method. So next, we wanted to look at assessing fraction metabolized. Fraction metabolized is the proportion of a drug dose that is metabolized by a specific enzyme or pathway relative to its total metabolism. So we use 2 low turnover reference compounds with known FMs as a proof of concept. These are risperidone, which is majority is metabolized by CYP2D6, and disopyramide, a majority which is metabolized by 3A4. So we did the knockdown using both the chemical inhibition method and the gene knockdown method with our clearance studies. And then the clearance of the parent compound, plus and minus siRNA or plus and minus inhibitor, over time was measured by LCMS. Looking at our first compound, risperidone, which is a low turnover compound, this does have a known in vivo FM of 70% for 2D6 and 30% for 3A4. It has been reported in publications that other in vitro models have underpredicted the FM for 2D6. Specifically in this graph here, you can see that the in vivo measurement was 77%, so around 70% FM. The green bar here would represent recombinant CYP enzymes. So you can see that the portion metabolized by 2D6 is underreported in this case. These are some optimized cell culture methods which researchers have prepared. And once again, we're still seeing underprediction there. In the top right corner here, you can see the specific study that we ran in HEPATOPAC with risperidone with both of our reaction phenotyping methods. On the left here is using the gene knockdown method. On the right is the chemical inhibition. And we can see in both cases, our controls, which would be our scrambled, in this case, you can see parent depletion. And also the orange here would represent no inhibitor, and you can see parent depletion. In both cases where we're inhibiting either by siRNA knockdown or by chemical inhibition, you can see there is a suppression in the parent depletion by the green line here or the pink line here. We did also in the study include stromal controls for both of these comparisons to show that there is no contribution from the stromal cells in this assay. Once we had predicted the clearance using these 2 different methods, we did then also do calculations for the FM. The FM was calculated using the siRNA knockdown method to be 0.7, and using the chemical inhibition method was calculated to be 0.89. We did want to point out that there is a publication by Smith in which HEPATOPAC chemical inhibition has been run, and they had a calculated FM of 0.83. And in this literature reference here, we did one which references in vivo FM for 2D6 for risperidone of 0.77. So in both cases, we were in line with the reported in vitro and in vivo data for FM in both of these cases for in vitro and in vivo, in both of our cases for the siRNA knockdown and for the chemical inhibition. Looking at our next example of disopyramide, this is another low turnover compound with a known FM of around 90% for CYP3A4. And in similar fashion, we did look at HEPATOPAC using both the siRNA knockdown method and the chemical inhibition method. In our control, so this would be no knockdown, our scrambled control, you can see parent depletion. Similarly, using chemical inhibition in the condition where there was no inhibitor, referenced by the orange line, you can see there is parent depletion. And in both cases, the green line here and the pink line here, you can see there is almost complete suppression of that 3A4 enzyme, and we're not seeing parent depletion of the compound disopyramide. And once again here, we did perform the hepatic clearance calculations and also compared it to the reported clearance in the literature. And we saw just 0.6 and 0.67 fold difference for the literature versus our observed in vitro clearance, and calculated our FMs as well. With the gene knockdown method, having an FM calculated to be 0.84, and using the chemical inhibition, an FM of 1. And once again, referring to the Smith publication, in which there's a number of compounds and a number of CYP targets that were looked at, the calculated in vitro FM in HEPATOPAC was 0.99 for 3A4 for disopyramide. And in conclusion today, we can state that the siRNA-mediated knockdown of CYP450 enzyme in HEPATOPAC is an effective and selective alternative to chemical inhibition for reaction phenotyping of low turnover compounds. Using the gene knockdown method with siRNA, we were able to achieve substantial and sustained reduction in gene expression and enzyme activity for the key CYP450s. We saw minimal off-target effects compared to chemical inhibitors. With the chemical inhibitors, we did see more broad, non-specific activity. And we did demonstrate that we were able to calculate the FM values with both methods, and they were consistent with published in vivo and in vitro data. This data does support expanding our capabilities in terms of additional CYPs and specifically for non-CYP targets, which will enable a broader application for drug metabolism studies. And we'd be happy to take any questions that you might have today, or you can feel free to contact us at a later date. My email is here, and of course you can always contact BioIVT via our customer service email address, and you'll be connected with the most appropriate person to help you with your questions.
All right. So we have now entered the Q&A session. If you haven't already, please post your questions in the questions pane. So let me check the questions pane, see if we have any. Okay, I have a question from Brian. He says, please discuss how you would interpret results with very high protein binding compounds greater than 99.8% that also have low turnover. So if something is more than 99.8% protein bound and have low turnover, how would we interpret the results?
There. Yep. So we haven't specifically yet, in terms of reaction phenotyping, worked with any compounds specifically with those types of characteristics yet. But we do typically, we have worked with clients just for clearance studies with HEPATOPAC that have had high binding protein capabilities. And we do work with both serum-free formulations and also serum-containing formulations to help work through different characteristics of compounds there. But we would be happy to speak with you further and bring in our experts on that topic as well.
I have a question. Are you planning to investigate any not CYP targets such as UGTs or targets without selective inhibitors?
Yep, that's one of the major goals, Seema, that we do have for the project. A lot of the interest that's helping drive this project was coming from researchers in the ADME space with low turnover compounds and who've been using the HEPATOPAC model. And so we had some initial interest with the CYP, looking at specifically the CYPs, but really what's been driving it is the UGTs and some of the non-CYPs. And so we had some experience knocking down the CYPs, so we were working through that protocol first. And actually our R&D team right now, one of our scientists is already working on the UGTs specifically there. As we're really finding from the feedback from the research community that this model might be helpful where in non-CYP cases and where there's not specific chemical inhibitors to selectively knock down the target of interest. And we're definitely interested in learning from people targets which may be of interest to them, so we can include them in our R&D efforts.
Thank you. I don't see any other questions, so I can kind of sneak in one more question, Jeannemarie, if that's okay with you.
Sure.
Have you applied this gene knockdown method in 2D cultures?
We have not specifically done it for reaction phenotyping. As was shared in one of our previous webinars, we have done a lot of work with HEPATOPAC with siRNA knockdown, not just for CYP, but also for disease modeling areas and GalNAc work. We've done a little bit of work, not for reaction phenotyping in 2D cultures in terms of siRNA transfection and GalNAc uptake, more with the GalNAc uptake, but for our work right now for the initial studies with the UGTs to see if we're targeting the right sequences, we are doing some work in 2D cultures before moving to HEPATOPAC.
Please note that we do have an array of HEPATOPAC resources available on our website. You visit the resources page and search for HEPATOPAC under education content. to find training videos, user guides, brochures, and applications. I think Jeannemarie mentioned that as well in her presentation. All of these is available on our website. And as a reminder, this is our final in our HEPATOPAC webinar series. If you missed the first 4, they are now available on demand on our website. Thank you very much for your attendance today. Here's my email address as well as Jeannemarie's. You're welcome to contact us directly. You can also contact us via the website at the address shown. Thank you again for your attendance, and I hope you all have a wonderful day. Bye-bye.